I'm Dr. Sayed Ahmadi, Technical Research Professor of Biology in the University of Chakwar. I say warm welcome to all participants in 7th International Conference on Applied Zoology 2024. And the topic that I'm going to discuss is about histopathology of heart and spleen as a result of chromium exposure in mice. The aim of study was to test the cardiac and splenic histopathology and microanatomical and morphometric changes at subtoxic chronic exposure in mice. And as you already know that chromium in our body increased risk of heart attack. Trivalent and hexavalent chromium cause a reduction of spleen in weight, splenocyte number, and percentage of blood lymphocytes. Chromium induces pronounced increase in the population of circulating RBCs or erythrocytes and also cause a reduction in the number of small lymphocytes. There is very little data available on the histopathology of chronic chromium exposure on the cardiac tissues. So, only a few studies have initiated or indicated that the effect of chromium on spleen histopathology. With keeping these findings in the mind, present study was designed. Experimental design was consist of 4 groups. First group was used as a control group. Normal drinking water was provided for the 30 days, and group 2 was chromium 50 ppm. Here, chromium solution 50 ppm was provided to animals on oral route, and group 3 was chromium 100 ppm. Here, 100 ppm chromium solution was given to the animal by oral route, and last route was chromium 200 ppm. Here, 200 ppm chromium solution was provided to animal by oral route. And then after dissection, organs were recovered like heart and spleen. Histopathological observation at 400x was done, and organ morphometry was carried out. Then the obtained data was analyzed through statistical applications like ANOVA and Dunker's multiple range test using SPSS 13. Here are the results. Here you can see in the first section, the selected sections of spleen in control, 50 ppm, 100 ppm, and 200 ppm. Here, number of splenocyte like red pulp cell, white pulp cell, and splenic cord cells were very much reduced as compared to control, especially in 200 ppm. While in the second section, here you can see the fibers of heart are in control, and then in 50 ppm, 100 ppm, and 200 ppm. Here, cardiac fibers were too much thickened and damaged, and plates were severely damaged, and fibrotic mass was created in the 200 ppm and 100 ppm as compared to control. Table 1 shows us the morphometric data of spleen and the heart, where histomatic parameters were measured like mean cross-sectional area of red pulp cell and mean cross-sectional area of white pulp cell, mean abundance, relative abundance of RBCs and WBCs in per unit area, and mean CSA of the cardiac fibers. The conclusion is that there is a clear impression of persistent low concentration 50 ppm chromium exposure in drinking water that can be dangerous to heart or myometrium, leading to the congestive heart problems, cardiac failures, bradycardia, along with many hematological implications. Thank you so much for your attention, and thank you so much for the listening.